I am Dr. Eram Hussain Pasha. I am from the Department of Biochemistry, and the topic that I will be teaching today is beta oxidation of fatty acids. Now, what is beta oxidation? So it's a major pathway of oxidation of fatty acids, and it is called so because fatty acids are oxidized in this process. Why is it called beta specifically? Because there is oxidation of the beta carbon of the fatty acid. Beta is the carbon next to the alpha, and the alpha carbon is next to the COOH group. So you've got the second carbon from the COH, that is beta. Now, where is it located? Where is this beta oxidation going to take place? It's going to take place in the mitochondrial matrix. What are the stages of oxidation that's going to happen? Number one, there is going to be activation of the fatty acid, because where is the oxidation taking place? It's taking place in the mitochondria. And where are these fatty acids moving about? They are moving about in the cytosol. So what you've got to do is you've got to transfer it inside the mitochondria. So for that, firstly it needs to get activated to a form which can be transported. Number two, after the activation, there is going to be transport from the cytosol to the mitochondria via a transporter. And number three is where the beta oxidation proper is going to take place, that is the mitochondria. So you've got three steps for beta oxidation: activation, transport, and beta oxidation proper. Now, let's start with fatty acid activation. Whatever fatty acid is coming and for undergoing beta oxidation, it's going to get activated. How is it going to get activated? The final product that is going to be formed is fatty acyl-CoA. Whatever fatty acid is there, that acyl-CoA is going to be formed. Fatty acyl-CoA. So how is that going to happen? By an enzyme known as thiokinase, and there is utilization of two high energy phosphate bonds. There is utilization of two high energy phosphate bonds, and the enzyme that is used is known as thiokinase. What is required in this? Coenzyme A is required and magnesium ion is required. Finally, fatty acid will be forming what? Fatty acyl-CoA. Coenzyme A is going to give the acyl-CoA, and then there is utilization of coenzyme A, magnesium, and two high energy phosphate bonds. Now what's going to happen? So the transport from the cytosol to the mitochondria is going to take place via a transport system, as fatty acid itself is impermeable. So what is the carrier system that is going to help? Carnitine carrier system. Okay, carnitine. It is a transport protein. Now, when we look at the transport that is going to happen, there are two enzymes that are going to take place. That is very important for this process to take the fatty acid in. Number one is CAT1, CAT1 also known as, and it is present in the outer surface of the inner mitochondrial membrane. Outer surface of the inner mitochondrial membrane. CAT2 is present in the inner surface of the inner mitochondrial membrane, IMM. Okay, one is present in the outer surface, CAT1 is present in the outer surface, and CAT2 is present in the inner surface. What is the full form of CAT1? Carnitine acyl transferase 1, easy. Acyl transferase, that is going to transfer, right? What is transferring? Carnitine is going to transfer. What is being transferred? Acyl group is being transferred. The fatty acid is being transferred. So the whole thing is known as carnitine acyl transferase 1, which is present in the outer surface of the inner mitochondrial membrane. Now you have carnitine acyl transferase 2, which is present in the inner surface of the inner mitochondrial membrane. Okay, this much is clear. Now what is going to happen? As soon as fatty acyl CoA is going to come in contact with Cat1, what is going to happen? The carnitine will form carnitine acyl CoA. Okay? What is going to form? Carnitine acyl CoA is going to form, and this carnitine acyl is going to get, acyl carnitine is going to get transferred inside the mitochondrial membrane. Okay? And on the other hand, what is going to happen? Acyl carnitine is going to again react with CoA, right? Because CoA was there present initially, and Cat2 is going to act on this. Finally, acyl CoA is going to get inside. What we started with was fatty acyl CoA, right? Fatty acyl CoA is combining with carnitine to form acyl carnitine, leaving out CoA that is present in the cytosol. Now this fatty acyl carnitine is going to get inside, then by the action of Cat2, it's going to react with what? The CoA is again going to come. Finally, carnitine will be separate, which will be recycled inside. And what is our end product? You have fatty acyl CoA. Mind you, the CoA, which is coenzyme A, which is present in the mitochondria, is very different from the CoA circulating in the cytosol. So, what is our helper in this? You've got the carnitine protein. Okay. Now, we get into the, once this is inside, the main thing that's going to start is the process of beta oxidation. How is that going to happen? You're going to have 4 steps. Oxidation, which is FAD dependent. Oxidation, that is the first step. Next is the hydrolysis, step number 2. Number 3 is oxidation again. But what is the coenzyme here? NAD dependent. It's going to be NAD dependent oxidation, and last it's going to be the cleavage, where acetyl-CoA is going to form. Now oxidation, it undergoes, what is the enzyme? Fatty acyl-CoA is going to undergo oxidation with the help of the enzyme acyl-CoA dehydrogenase, and it is FAD dependent. What is being formed? Enoyl-CoA is formed. This enoyl-CoA undergoes hydrolysis again by enoyl-CoA hydrolase, and where there is taking up of the H2O, and what is formed? 3-hydroxyacyl-CoA. This 3-hydroxyacyl-CoA is further, I said the third step is going to be oxidation again, so you know there is a dehydrogenase that is working. So what is the dehydrogenase? 3-hydroxyacyl-CoA dehydrogenase. This is NAD dependent, so there is utilization of NAD, and then what is formed? 3-ketoacyl-CoA. 3-ketoacyl-CoA then finally undergoes cleavage, where the fatty acyl-CoA and acetyl-CoA is separated, and the enzyme is thiolase. So by the end of it, you have the removal of acetyl-CoA and rest of the fatty acid. So let's revise. So you have oxidation, hydration, oxidation, cleavage. The first oxidation is FAD dependent. The second oxidation, which is in step 3, that is NAD dependent. The enzyme are dehydrogenases, okay, and there is utilization of FAD and NAD in both. Okay, then you have hydration, hydratase, that is the second step, and then you have the thiolase, which is the cleavage step. So by this, supposing the fatty acid is a 6-carbon fatty acid, supposing. So the first step is removal of 2 carbon, that is acetyl-CoA, then you will be left with 4 carbon fatty acid and acetyl-CoA. Then this step will again take place, the oxidation. Then what is going to happen? Again 2 carbon acetyl-CoA is going to get separated, and so you have in total 3 acetyl-CoA, which is formed from a 6-carbon compound, and the whole the whole oxidation will be taking place twice, thereby forming 3 acetyl-CoA. Okay, so you have to understand that when you have an even chain fatty acid that is going to get oxidized, so the number of acetyl CoA will be divided by 2 because 2 carbon are going to take place, and the number of times the cycle is going to take place will be a number less. For example, you have a 8 carbon fatty acid that is going to undergo oxidation. So the total number of acetyl CoA will be coming out will be 8 divided by 2, that is 4 acetyl CoA, but the cycle will not take place 4 times, but the cycle will not take place 4 times, it will take place only 3 times. Okay, so with that we come to the end of the fatty acid oxidation. Thank you.